Hello everyone, this is Professor Vijay Lakshmi from School of Pharmaceutical Sciences, VISTAS. In this session, you will understand the principle of HPTLC, instrumentation, and step-by-step procedure from sample application till analysis of the phytoconstituents. High-performance thin-layer chromatography is the advanced and automated form of TLC, which uses pre-coated plates with finite particle size of about 5 to 6 micrometer to achieve sensitivity and reproducibility of the analysis of phytoconstituents. HPTLC also provides high resolution, reproducible and automated analysis. These are the various steps involved in HPTLC analysis. It starts with sample preparation, selection of chromatographic layer, selection of plates, application of the sample, selection, optimization of the mobile phase, chromatographic development, detection of spots, and scanning and documentation. HPTLC is based on the principle of adsorption chromatography, in which the components of the samples are separated according to their different affinity towards stationary phase and mobile phase. First step to extraction of the materials using suitable solvent and selection of suitable extraction methods. After extraction, the extract should be filtered using suitable filter paper like Whatman filter paper to remove the insoluble particles. Then the filtrate can be concentrated under reduced pressure using rotary evaporator, and we can reconstitute in a suitable solvent before application. The sample should not be excessively concentrated as overloading can cause broad bands and poor resolutions. After preparing the samples and standard, the next step is the selection of stationary phase. The choice of stationary phase is primarily based on the polarity and chemical nature of the compounds to be separated. Silica gel 60 fluorescent 254 is the most widely used stationary phase in HPTLC. When conventional silica does not provide adequate separations, specialized phases like RP18, cellulose, diol, or amino phases may be selected. HPTLC plates is usually made of glass, aluminum, or plastic and coated with a thin layer of adsorbent. And these pre-coated plates are available in different sizes like 20 into 20 centimeter, 10 into 20, 10 into 10, and 5 into 10 centimeter. The plate size is selected according to the number of samples, and silica gel 65 F254 is the most commonly used plate for pharmaceutical analysis. Here the fluorescent 254 indicator allows visualization of UV absorbing compounds at 254 nanometers. And the make sure that the plates should be handled only by the edges, avoid touching the coated surface, and protect the plate from moisture, dust, and contamination. And before proceeding, we have to wash the plates because there is chances for the absorption of moisture and also volatile impurities. So by using the solvent like methanol or combination of solvent chloroform methanol, we can wash the plate. Any of the three methods can be used. For ascending method, the plate is placed vertically in a chamber. The solvent ascends carrying impurities to the edge. And in dipping method, it is a rapid method in which the plate is immersed into the trough containing the solvent. And in continuous method, the solvent flows continuously over the plate for fixed durations. Next step is activation of the plate. As silica gel is hygroscopic and it can absorb moisture from the atmosphere, excess moisture can alter the interaction between the analyte stationary phase and mobile phase, which results in variation of RF values, band shape, and reproducibility of the result. So activation involves controlled heating of the plate in an oven at a temperature between 110 to 120 degree for 30 minutes. to remove the adsorbed moisture from the stationary phase. So next is the sample applications. So in Camag HPTLC, we'll be having a applicator, Illomet applicator, and the syringe whose volume is about zero point 0.1 ml, that is 100 microliter. So first load the solution into the syringe, fix a pre-coated plate properly in the applicator, and define the application positions and track spacing. Apply the record volume as a narrowband. The sample should not be overloaded because excessive sample concentration may produce broad bands and poor resolution. The next step is the selection of mobile phase. The selection of the mobile phase is a critical step in HPTLC method development. So mobile phase is selected based on polarity of the analyte, chemical nature of the compounds, stationary phase, etc. For polar compounds, the mobile phase may contain a combination of non-polar, medium polar, and polar solvent. But the exact ratio must be optimized experimentally for the particular sample. The next step is the development of plate, and different shaped developing chambers is available, like a twin trap chamber, rectangular chamber, V-shaped chamber, sandwich chamber, horizontal development chamber, and automatic development chamber. So before development, the developing chamber is saturated with the mobile phase vapor. So the mobile phase is added to the chamber and the chamber is closed. This saturation will help us to establish equilibrium between the mobile phase and chamber atmosphere, and it produces uniform solvent movement and minimize the variations in RF values and improve the separation. You can also line the chamber with filter paper if required to facilitate saturation. So chromatographic development and drying. After sample application, the HPTLC plate undergoes chromatographic development. The mobile phase moves upward through the stationary phase by capillary action, during which the components of the samples are separated based on their different affinities for the stationary and mobile phase. Once the solvent reaches the predetermined development distance, the plate is removed, and after development, the plate contains a residual mobile phase and must be removed by drying before reduction. Avoid using hair dryer as there is chances for the evaporation of essential oil components. Densitometric scanning. The developed HPTLC plate is scanned using a scanner at a selected wavelength. The scanner measures the intensity of absorbance, reflectance or fluorescence of each separated band. The scanner moves across the tracks and measures the optical response of each separated band. The measured signal is converted into a densitogram in which we can see a peak. Each component appears as a peak. And the scanner consists of light source, optical system, monochromatic system, detector, and computer and software. Modern systems can scan the plate in absorbance or fluorescence mode at selected wavelength. The developed plate is positioned correctly in the HPTLC scanner. So we can position to check the correct position. We can see the green colored spotlight on the tracks. So then start the scan the tracks. Then the scanner moves across the selected tracks and measure the optical response of each separated band. The result is presented as a densitogram. Here that it consists of a graph which consists of x-axis and y-axis. X-axis measures the distance and y-axis absorbance. So the area of height of the peak can be used for quantification. Detection and visualization of spots. The separated components on the HPTLC plate are detected and visualized by identify the individual spots. Detection can be performed by direct visualization or But that with derivation followed by visualization. So the developed plate can be examined under different wavelengths like 254 nanometer, 366 nanometer, also under visible light. In 254 nanometer, the compounds absorbing 254 nanometer will appear as a dark zones against a fluorescent background when you are using silica gel F254 plates as a stationary phase. Under UV 366 nanometer, some compounds exhibit natural fluorescence and appears as a colored fluorescent band. This wavelength is particularly useful for detecting compounds that are weakly visible at 254 nanometer. And the compounds that are naturally colored can be directly visualized under visible light. And the colored pigments like anthraquinones and phenolic compounds can be visualized under visible light. The next step is derivatization. Some phytoconstituents have weak absorption in UV. In such case, the plate is treated with a suitable derivatizing agent. So, for example, for terpenoids and steroids, we can use anisaldegate sulfuric acid as a derivatizing agent. For alkaloids, drag and drops reagents, and for phenolic compounds, we can use ferric chloride reagent. So, we can derivatize the plate by placing the plate in a automated reagent spraying in which the detecting agents are sprayed uniformly over the plate. Also, we can dip the plate in a detecting agent placed in a chamber. And for volatile components, we can use the thermal activation. So, at the temperature on heating this plate, we can able to visualize the terpenes and terpenoids present in volatile oils. The next one is a quantitative analysis. So, for quantitative analysis, a calibration curve is prepared using different concentrations of the standard. So for this, prepare a stock solution of the marker compound of known concentration. Usually 100 mg per ml will be the stock solution concentration. Then prepare a suitable series of working concentration and apply the different standard concentrations and sample solution as a narrow bands on the same plate. Develop the plate using the optimized mobile phase under controlled condition. Detect the marker compound at the optimized wavelength and after suitable derivation. So after chromatographic development and detection, the plate is scanned densitometrically and the peak response of peak area is recorded. So a calibration curve is constructed by plotting the concentration of the standard against its response. So the response obtained from the sample is substituted into the regression equation to determine the concentration of the marker compound. Appropriate dilution and sample preparations factors are then applied to express the final amount of analyte in the sample. So this is a WinCat software which is used for analysis of these phytochemical extracts in HPTLC. So in the left corner we will find out step-by-step procedure. So first you have to select what type of analysis we are going to perform, whether qualitative or quantitative. So if these two options will be available there, we can click either qualitative or quantitative. Then the next step is selection of the stationary phase. So we usually, as I said, silica gel 60 fluorescent 254 will be the standard stationary phase commonly used. And we have to select the solvent which is used for washing of the plate. Then we have to load the micro syringe with the sample and we have to enter how many spots we are going to spot on the plate and the volume of the spots also we can enter into the software. Then we will have like this layout of applications. So based upon the number of samples we have entered in the software, we will get like this layout. And once you have placed the plate on the applicator and entered, we will start application of the sample. Starts with the last spot. Once the spot is applied, automatically it will start applying the next spot. And if you want to change the sample means again we have to refill the syringe. Before that we have to wash every time with methanol. Then fill with a suitable test sample and start the procedure. So after scanning the plate, we used to get a peak like this, and we have to assign the peak, and we have to check for the presence of this marker compound in all the peaks. For example, if you are using gallic acid as a standard, and if you have selected that gallic acid, we used to check the presence of this gallic acid in all the test sample. This is the 3D chromatogram of all the tracks. So once you have assigned the marker compound, we will get a graph like this. So this is an example of using gallic acid as a marker compound. Once you have assigned the chromatogram, we can see all the presence of this gallic acid in all the test sample in different tracks. This is the calibration curve for that standard compound. Then we can get the quantification of the compounds based on the graph height and graph area. And this is a fingerprint of that analysis. Then coming to the applications. So this HPTLC is most commonly used for both qualitative and quantitative analysis of phytochemicals present in the herbal extract, in the dietary supplements and nutraceuticals, and for the estimation of pesticides or coloring substances in food analysis. HPTLC is used to detect, also used for detection of cholesterol in edible oil. And in cosmetic analysis, it is used to detect the presence of traced materials or prohibited substances, if any. And in pharmaceutically, it is used for the detection and identification of raw materials and metabolites in the biological media. And in clinically, it is used to study the metabolites in all the drug in the samples like plasma, blood, etc. And to investigate the poisoning case, it is used in forensic applications. So the advantage of HPTLC, it is simple and rapid technique, relatively requires low solvent consumption. Multiples of samples can be analyzed simultaneously. It is suitable for herbal extracts, provides both qualitative and quantitative analysis, and requires relatively small sample volume, and useful for getting fingerprinting, and it is allows visual documentations of all the compounds, and useful for batch to batch studies. So thank you for your patient listening. Thank you.